Hello, everyone. Welcome to a webinar on data mining and its application in epigenetics. I'm pleased to welcome our two speakers of the day, Damien Calay and Martin Seifert. Damien, yes, thank you so much. So Damien graduated from the University of Namur, where he got a PhD in cellular and molecular biology. He then spent 6 years doing research at the Imperial College London. Damien's skills and growing interest for multi-omics data analysis took him to Diagenode, where he is supporting scientists across the globe with advanced bioinformatics data analysis. Prior operating companies active in the bioinformatics and data mining, Martin was an assistant professor at the Technische Universität München, expert in microarray analysis. Martin has published more than 50 peer-reviewed papers, and he's also the co-author of 2 patent applications for the diagnostic of prostate cancer. He's passionate about precision medicine and how omics technologies can translate into clinical settings. Today, Damien and Martin will talk about the utility of data mining in the discovery of patterns in our field of interest. They will take your question at the end of the presentation. You can ask them in the question and answer section. If you have any technical problem, please use the chat for that. So Damien and Martin, the stage is yours.
Right, thank you very much, Jerome. And hello everybody, and thank you for joining in. Thank you for the nice introduction. So here is the content of the webinar today. So I will first introduce you to some concepts and definition about data mining and machine learning. Then I will explain a little bit what is machine learning doing. So it's really pattern discovery and classifier generation. I will list a few application areas, and then Martin will go through a case study of classifying cancer tissue versus normal tissue using DNA methylation data. So let's start with a few concepts and definition. What is data mining? So it is actually the process of identifying informative features or patterns in very large data sets, what we call big data. And it's involving multiple methods, which are basically a combination of statistics, artificial intelligence, and machine learning. I will go a little bit more in details about some of these concepts, starting with what is called big data. And it refers to datasets that are just too large or too big and too complex to be analyzed by traditional data processing software. And in our day-to-day life, we have examples of big data. So we have the New York Stock Exchange, for example, which generates about 1 terabyte of new data every day. On another level, we also have social media. For example, Facebook database. It can generate up to 500 terabytes of new data every day. And in the biological field, all next generation sequencing technologies also generate big or very big data sets that we consider as big data. They are in a way kind of structured, but they're just too large. On the other hand, we can have data that are too large as well, but also unstructured. What I mean by this is that it's coming from heterogeneous samples, like for example, a human cohort study, where you have metadata on top of your epigenomic or genomic data. These metadata can be clinical data, epigenomic or genomic data, transcriptomic data, lifestyle data, any kind of other data. And so dealing with this kind of big data and drawing conclusions from this, it requires a computational analysis that cannot really be achieved with standard statistics. And what I mean by computational analysis, it means artificial intelligence, which is intelligence demonstrated by machines. And this term is actually used to describe the computers that can mimic the human behavior. So they're trying to mimic the functions that are usually associated with human minds, such as learning and problem solving. And we have examples of artificial intelligence where it's used. For example, the Apple artificial intelligence Siri. It's also involved and used in online game playing, intelligent humanoid robots, and self-driving cars. And a subset of artificial intelligence is machine learning. And machine learning relies on algorithm and mathematical models that the computer will use to create knowledge from past experiences. And it will do this without being explicitly programmed to do this. This relies on patterns, and that's really a critical word in machine learning, is patterns. And it will use this pattern not only to analyze the data, but also to make predictions about the future. Because the machine learning algorithm will build and learn from past experiences to identify patterns and make predictions. So there are three types of machine learning. The first one is supervised, where the machine will learn from a training data set. So there will be an example of this supervised machine learning just afterwards. And so in this supervised machine learning approach, we are using a subset of data to identify patterns and create a classifier that will be used to analyze another data set. The second type of machine learning is the unsupervised machine learning, where no training data is required. And that's mainly when we are talking about clustering or principal component analysis. And the third one is reinforcement, where a machine learns on its own, and that's more related to the artificial intelligence part. When machine learning is particularly helpful is when we are dealing with what we call wide data, in contrast to long data. So wide data refers to a data set where we have actually many variables in comparison to the number of subject or samples. And examples of this wide data can be when the data sets are originating from multiple sources. So if you are combining phenotypic data like age, gender, with genomic or epigenomic data, this can be, for example, the presence or absence of a SNP, any sort of clinical data, the recurrence of a disease, for example. So that will create a huge amount of different variables for each individual or sample that is to be analyzed. Another example, on the other hand, can be that the data set is coming from a single source, but it includes many variables. This, for example, is the Infinium DNA methylation epic array, where you have 850,000 probes looking for DNA methylation of specific CPG. So you would have 850,000 variables, values for each sample or individual. When we are talking about epigenomics, for example, RRBS in human samples, we can detect on average 4 million CPGs, meaning 4 million variables. That's also considered, of course, as wide data. And we can also deal with other epigenomic technologies like ChIP-seq, ADAC-seq, or WGBS data that would all be considered as wide data. Of note is to see the differences between machine learning and artificial intelligence. And I would like to point just two differences, main differences here, is that artificial intelligence is really trying to mimic the human behavior and perform any task like a human being would do. So it's really trying to do some reasoning. While on the other hand, machine learning is built to really perform a particular task, a specific task, and will try to be as accurate as possible on doing this task by learning from past experiences and building patterns. And a question often arises when we are talking about machine learning is, why would I use machine learning over standard statistics? So there are multiple differences between these two approaches. One of them is that machine learning is more suitable for large and or complex data sets, as said previously, as statistics is more suitable for small or less complex data sets. And also, while statistics relies on mathematical model and draw conclusions only based on evidence, machine learning will rely on mathematical model and algorithm, not only to draw conclusion, but also to build a pattern that will be able to predict future events. So what is machine learning actually doing? So it's using algorithm to find informative features, what we call patterns, to build predictive classifiers from complex data. And here is an example of a supervised machine learning approach where we have a human population and we have, for example, DNA methylation data for this human population. We are selecting a training cohort. So I forgot to say that in this population, we have healthy or diseased people. So we are selecting a few of them where we know the state of these people, if they are healthy or diseased. So that's considered as our training cohort. We feed the DNA methylation data of this training cohort into the machine learning environment. We will apply different algorithms, for example, simple logistics or random forest, to find informative features that will build a classifier to discriminate healthy from diseased people. And then, because this is a supervised machine learning approach, we can use the classifier that was built from the training cohort to determine the health status of the rest of the population based on the DNA methylation data, for example. So that's really the main concept, and the key concept of machine learning is really creating a classifier that will be as accurate as possible to discriminate two populations or two categories of samples based on patterns. Just a quick word on application areas and which kind of data would you need to do some sort of machine learning approach and data mining through machine learning. So if you're interested, for example, in biomarker discovery, if you have all sorts of not only single source data, but data coming from multiple sources, it can be genomic data, epigenomic data, also clinical data. You can use machine learning approaches to really find informative features inside complex data sets. It's also interesting in interactomics. So when you have multi-omics data, so you have the integration of epigenomics data with transcriptomics data, metabolomics data. So overall, this is very complex and very hard to find informative features just with standard statistics. And really, machine learning can help in this kind of situations. As said previously, also if you have data from clinical studies on human cohorts, for example, you usually have data coming from multiple sources, a mixture of data that can be clinical, that can be lifestyle data, genomic data. And so again, you would have data that are so large and so complex and coming from multiple sources that it's difficult to deal with standard statistics. And finally, the last example here is single cell analysis, because for each single cell, so it can be single cell RNA-seq or single cell ATAC-seq, for example, you would have information on the expression level or the open chromatin regions for each single cell of your sample. So you would have a lot of variables and a lot of samples, because each sample would be basically one cell. And so dealing with this kind of data can be quite tricky, and that's where machine learning can help a lot. So just to summarize the type of data that can be fed into the machine learning environment, we can deal with all sorts of genomic, epigenomic data, multiomics data, complex data in general, coming from a single source or multiple sources, data from clinical trials, and single cell analysis data, environmental data. So that's it for my part. Now I will let Martin maybe go through the case study, which we built by classifying cancer tissue versus normal tissue using DNA methylation data.
Yeah, thank you very much, Damien. And hello, everybody, also from my side. And I just try, now I have control of the screen, perfect. Yeah, as Damien just said, I would like to make you familiar with the machine learning approach and the data mining approaches we are applying with a practical example, and this is classifying breast cancer tissue versus normal tissue. And I just also would like to point your attention towards a white paper which is available at the Diagenode website also for download, which also summarizes what I will tell you in the next couple of minutes actually. So what is the data set about? Well, the data set is a DNA methylation data, or better to say two data sets, two DNA methylation data sets, which were produced using the Illumina Infinium methylation 450K arrays. Both data sets are published and are available via the Gene Expression Omnibus, and you see the GSE numbers up here. And the first data set was derived from a paper which was titled The Integrative Epigenomic Transcriptomic Landscape of ER-positive Breast Cancer. And this is a data set which contains in total 313 samples. And as we were looking for data which allow us to compare healthy specimens from disease specimens, we took 50 from specimens and data from healthy women here. And out of the 263 breast cancer samples, we randomly just selected 52 out of this data set. I would like to mention that this data was done by mainly an international group, actually, with people from China, from Shanghai, from the University Clinic in Erlangen, but also and mainly also from the University College of London. It was published in Clinical Epigenetics in 2015. I hope you can hear me, because I'm getting a sign that my audio connection has an issue.
I think well, the sound was not that good at the beginning, but I think it's getting better.
Okay, all right. So I hope that works. Just, Damien, if you, okay, perfect. If just somebody raises a hand if you should have any issue in hearing me. The second data set contains 57 samples, 40 primary breast tumors, and 70 normal tissues. So what we have here are two completely independently created data set. One, as I said, mainly from the UCL, and the second one is coming, should also mention that, from a group in Barcelona. And it was published also 2015, but completely independently in Molecular Oncology. So what did we do? We just followed an approach which is quite typical, and Damien mentioned that before. We used a supervised approach in this. And what we are striving for is to find informative patterns and to build a classifier which allows us to distinguish normal from breast cancer tissue. And in this case, we are very lucky that we have these completely independent two data sets, because we can use the first one for the training of the system and building the classifier, and we can use the second data set then for evaluating the classifier and applying it completely to a completely independently derived data set and see what comes out from this analysis. So what we did here, or what I will show you, is that we loaded the data set 1 into the machine learning environment. We carry out the machine learning, build the classifier, and apply the classifier to the second data set. So for this specific analysis, we use a very handy and practical machine learning environment, which is called VECA, which is provided by the Waikato environment. Well, this is the Waikato environment for knowledge analysis, and is provided by the Waikato University in New Zealand. This can be used freely for everybody, and it's a very handy and versatile tool to do machine learning. So what we did here, we put the data set 1 into this environment, and you can see it here on the right hand side. As I mentioned before, we have 50 normal samples in here, and we have 52 breast cancer samples in here. That's also depicted in these colors here. And what we have from the 450K arrays are the 485,513 CPGs, which are in principle then here also depicted in this window. And it is exactly what Damien mentioned before. It's a so-called wide data set, which has not too many different samples, but still a huge number of different features here. So we let it run, and we choose mainly two different approaches. One is called simple logistics. Damien mentioned that before as well, and the second is called random forest. And I show you mainly the results of the simple logistics. The random forest delivers very similar results. That's just something I would like to mention. And what you can see from this results picture, after we did the training and the internal evaluation within the first data set, we can see that we can identify, or better to say, correctly classify 95%, above 95% of the instances, meaning correctly classifying normal and breast cancer tissues. So what we have are 97 samples out of the 102 were correctly classified as control or as breast cancer tissue. And this then is also depicted in this confusion matrix here. So we have from this here, from the 50 normal samples, we have 48 classified as A, which means normal, and 2 as breast cancer. And we have 3 which should be actually breast cancer as normal, but 49 are correct. So this is a very good classifier. With above 95%, we can do the classification correctly within the first data set, I have to say. This is the training data set with the internal evaluation. So what does the classifier then look like? Well, at the end of the day, the classifier is made out of 16 CPGs within this data set, And these 16 CPGs are informative features which allow them to identify and to predict whether a sample belongs to breast cancer or normal tissue. And what I have depicted here on this slide is, in principle, the methylation degree of normal and breast cancer, normal in blue, breast cancer in red. And these are the 16 CPGs, which you can see here. And I just pull out one, a little bit bigger, or two better to say, and you see that in a normal tissue, this specific CPG is relatively low methylated for normal tissue and relatively high methylated for breast cancer. And for the other one, which I just picked here, it is the opposite. It is relatively high methylated into normal tissue and relatively low methylated for breast cancer. What I would like to show you here with these two examples is that from a statistical point of view, the data is still heavily overlapping between normal tissue and breast cancer tissue. And this is true actually for all these 16 CPGs. They are all more or less, from a statistical point of view, as single features not significant. But if we then do a combination of the 16, the classifier still is able to predict breast cancer from normal tissue with more than 95% of accuracy. And that's also one of the difference I would just like to mention. It is something different whether you do statistics, say, okay, this is a significant CPG, or this is a part of a classifier, an informative feature, which in total then builds the classifier. If we go one step further, and data mining is not only the machine learning part, but also bringing all into context again, we just use the 16 CPGs here and have a look where they belong to, and have here a list of gene symbols of these CPGs, which you can see here. And we have, in addition to that, projected them to a literature network actually, and tried to figure what the meaning of these combinations of the 16 CPGs then projected to gene symbols would be. And you see the network on the right-hand side here. And what you can see quite obviously is that a lot of those are actually also connoted as, for example, malignant neoplasm of breast, or other breast carcinoma. And you have candidates in here like NCO2, SOX1, carcinogen kinase 2A1, and so on, which are all well known to play a role in breast cancer mechanisms. So that is then an additional layer which you can add to the pure machine learning part to bring your results into biological context again and learn more about what the meaning of the different informative features are and what your classifier is in principle all about. And of course, if you can do this kind of projection and find out things like this is all very strongly associated with the mechanisms you are looking at, then this is very assuring that your classifier is not giving you results which would be some incidental finding or whatever. It is giving you a very clear indication that you're looking into the right direction. So last but not least, of course, you can say, well, this is more or less could be a self-fulfilling prophecy. You did the analysis within one data set, but can you generalize that, or what would it mean if you have an independent data set? And therefore, in this case, we have this very nice second data set, which we use for cross-validation. So we uploaded the second data set also into Weka and did not do any new training or so. We just used the classifier from the first analysis, from the first data set, and applied it. So that is our validation approach. We take data set number 2, apply classifier from data number 1, and have the evaluation layer number 2 in this. And what comes out here is very interesting. This second data set, which is slightly smaller than the first one, still is giving us results with about 95%, to be correct, 94.73 is the number for the percentage of the correctly classified instances. So we have only 3 not correctly classified, and you see here also the confusion matrix. The normals are all correctly classified, and for breast cancer we have 3 which out of the 40 which are actually not correctly classified, but it's still a very, very good result. And just bear in mind, these are completely independently derived data sets produced from 2 independent groups in 2 different laboratories, and still the classifier works extremely good on that. So what we have here is a very good confirmation that this classifier can be used to really predict breast cancer from normal tissues in completely independent data set based on the DNA methylation from 450K arrays. So as a short summary for this example, and one thing I really would like to mention is this is a very straightforward approach which we used here, and there are many data sets which can be used in this in these situations, and it's really not too complicated and can bring very nice results. So what data mining using machine learning gives us here is a straightforward approach to address complex and or big data sets in a very efficient way. It offers great opportunities for the analysis of complex disease situations when we can combine it with phenotypic and other omics data. In this case, we have, from phenotype perspective, we have only diseased, non-diseased, which is working very straightforward here, but in other situations, we very often have also additional other data, which we can very easily also integrate in our analysis strategies. So we use algorithms to identify patterns and build classifiers, and we use the classifiers very simply then to categorize and see and predict in principle the individual samples. And last but not least, it can be very helpful to have also a biological mining layer at the end of the analysis or somewhere in the process of the analysis, which allows also for biological classification and having a look at biological mechanisms. This can be very reassuring that you are on the right track and that what you find as a classifier is in conjunction with actually what you are looking at from a biological perspective. So we are giving this talk, of course, today also to make you familiar with our offering at Diagenode, and this is, of course, something which we provide as a service for you. We integrate the data mining into the other service aspects Diagenode is already offering. And what we can say is that we have a proven track record in especially the epigenetics environment, in chromatin and DNA methylation analysis, but of course as well in RNA-seq analysis. We have good expertise in the bioinformatics aspects and of course also in data mining. And what I also would like to mention is that the services and the data mining service is fully integrated with the other Diagenode services, so it's a service which can run from A to Z. It is efficient and reliable because what we try to do, and I hope Damien and I were able to show you that, we try to do it very straightforward without becoming too complicated. And this is also then at the end reflected in a comprehensive reporting which we provide. And of course, one of the key aspects in all these services, and especially the data mining, is a close communication with you, with our customers, to understand and completely comprehend the data sets we are looking at. And with that, I hope we could show you a little bit of what we are doing in the environment of data mining. And I would like to thank you. And of course, we are open to your questions now. By the way, more information you can also find on this email here, which is depicted here.
Yeah. Mm-hmm.
Thank you very much.
Thank you for this very informative presentation, Damien and Martin. We're ready to take a few questions, if any. We got one regarding the methylation analysis of breast cancer sample.
Mm-hmm.
Just a comment about the methylation site on the y-axis. If you can give a little more information about the information that is present there on the graph. It's slide number 23.
Do you want to go back, Martin, or
where we're comparing, breast cancer mutation with
this one, I guess.
here, what you can find on the y-axis.
Yeah, that's in principle the beta value, the methylation. So that's the if you take this as a this is ranging from 0.087 degree of methylation to 0.72. So that would mean 72% of methylated sites. And these are the symbols here. In principle, these are the different samples from normal and from breast cancer. And you have the same here. That's just the left-hand side is normal and the right-hand side, the red ones, is breast cancer. Does this explain?
Yes.
So this goes in principle from 0 to 100%. But as in biology, very often it is not 100% demethylated and 100% methylated, but that's the range which we can find here.
And another interesting question, what is the limit in number of samples that are required to do this type of data mining analysis?
Yeah, that's a very good question. Because very often people think you need thousands of samples, but I'm very much convinced also from many examples and many analyses which we have done that a reasonable number starts also relatively low. Low means 20 per group can be sufficient. Sometimes even only 10 per group can be sufficient. This depends then on, at the end of the day, also a little bit on how strong the effect is. Of course, the more the merrier, the more samples you have, the better it is. But it is what we are providing here is not, many people think that machine learning and AI is always deep learning and is neural networks and so on. And of course, therefore, you need thousands of samples to come to a classifier. Applying machine learning algorithms like simple logistics or random forest and so on, you can start with numbers in the range of 15 to 20 samples per group.
Just to comment on that as well. So we did an analysis starting from around 50 samples per group, and we built a classifier. And by lowering down the number of samples per group, when we reach around, as Martin said, around 20, we were still able to have a classifier that was able to predict approximately with the same accuracy as with 50 samples per group. So you don't need actually many samples in each group of samples to do data mining.
Yeah.
And to build a classifier which is actually very good.
Damien, Martin, we have another question, practical one. Can you deal with different number of samples if you have, different groups? So meaning, for example, 10 or 15 sample in a cancer.
Absolutely.
Yes.
Yeah.
Yes. Yes. And I've shown you the analysis stream today in the way that we did dataset 1 first and then dataset 2 as validation. We did it vice versa as well, and there you have an unbalanced data set. In principle, if you take dataset 2, we have more cancer samples and less normal tissues, but it still works also the other way around, actually. So you can have unbalanced numbers, but of course, also your, the lower number should be, in the range of somewhere 10 to 20, but it's not, you cannot say it from an absolute general perspective. It is really also depending slightly on the strength of the signal you'll be able to find. But we always do, within the training phase, we do internal cross-validation approaches. And this is very, very good to get an estimate if the signal you're retrieving is valid or not.
Excellent. And so we'll just try to take a few more questions we got. So regarding, the study, why only 16 CPG were used? And another question is also related to that. In the analysis here, if we see also minimal change, in the methylation level, or can we use that in the future, for example, to detect change in methylation in cell-free tumor DNA in blood?
Yeah. So the first question is easy to answer, because simple logistics delivers us these 16 CPGs as being informative for classifying breast cancer versus normal tissue. So this is within the algorithm of the simple logistics, which we used as machine learning algorithm here. These 16 CPGs are those which are delivered, and there may be still some others in there, but by applying that algorithm, that's the classifier. And that's done iteratively in the process of the machine learning. The program tests out different settings, and this is then at the end the best setting which we can find for classifying breast cancer versus normal using this specific algorithm.
Thanks a lot. And the last question before we go.
The second question was, well, you can use this, of course, such patterns also then for classifying other sample types for cell-free DNA and so on. And actually what is done very often now is that these patterns and also these predictions can be used also for building biomarkers for liquid biopsy, etc. Sorry, Jerome, I didn't.
No. Thanks a lot for the very detailed answer. So maybe the last before we go, is there any kind of pattern in the wrong calls that are made by the classifier? Or that's not something, of course, we can, identify? I don't know if you got the question right,
Good question. If there is something specific in those which are wrongly classified. Mm-hmm. Sometimes that's the case. In this case, we haven't looked into that, because that's only a very low number. But if you would have a higher number, that is something, sometimes you can look into that, because that could be something then producing some confounding aspect or so. But in this case, we haven't looked into that.
If we have the information about metadata, there might be
exactly
something into this metadata, like a clinical feature that can explain why maybe 2 or 3 samples were incorrectly classified.
Yes.
Okay, thanks a lot. Thank you all for joining the webinar. Of course, we remain available if you want to pursue the discussion. Just go to our website, reach out to us, and we will be more than happy to continue this very interesting data mining discussion. I wish you a great evening or day, and looking forward to be in touch in the future.
Thank you very much, everybody.
Thank you very much. Bye-bye.
Bye.
Bye.
You're still in control of my computer, Martin.
I just tried to release it.
Oh, yeah.
Now you should have it back.
Yeah, I have it back.